Hi, my name is Julia and I'm a third-year biomedical student at the University of Windsor working in Dr. Hallman's micro-nano lab. My research is basically studying the science of cell squeezing. Imagine going to a grocery store to shop for tomatoes. By grasping the tomato, you can distinguish its quality based on the mechanical properties, the firmer tomato being of optimal quality. At a smaller scale, biological cells follow a similar analogy. When a cell is squeezed, a healthy cell will give a different mechanical signal than a diseased cell. Squeezing cells at such a small scale requires miniature squeezing devices, since the average size of a human cell is about 3 times thinner than the width of a hair. In Dr. Ahmed's group, we are innovating miniature squeezing devices based on 3-dimensional printing. These tiny devices can function as a complete diagnostic lab to detect diseases and to understand how a cell functions under confinement. In doing so, we are able to detect the first signs of complex diseases, such as cancer, by simply testing the mechanical properties of cells, one cell at a time. The microfluidic devices can allow for a cell to be squeezed within a 3-dimensional space. Molecular architectures of cells are based on its cytoskeletal components, which determine the overall deformability and mechanical response to transduction. Any change to the cell, such as disease progression, can significantly alter its mechanical properties. For instance, it is known that as a cancer cell progresses, its cytoskeletal proteins transform to increase its ability to deform and contract, which results in increased mobility to travel to various tissues. This process is known as metastasis. By understanding these physical differences between a cancerous and non-cancerous cell, the lab-on-a-chip device can be designed to isolate the cell and to mechanically transduce it to release its contents for further analysis. My focus is to apply the known properties of cancer cells to a reconfigurable lab-on-a-chip device using computer-based simulations in order to determine the mechanics of the chip required to identify the specific characteristics of single cells. Ultimately, the end goal for lab-on-a-chip research is to eliminate the ambiguity and invasive nature of current cancer testing measures, to provide a more effective point-of-care alternate. My role in engineering the chip to detect single cells is only the beginning for the future of lab-on-a-chip research. I look forward to sharing more about our research in the upcoming conference. Thank you for listening.